Hi, this is Kristen Atchison, and we're still talking about the brain and behavior. Before we get into discussing the actual brain itself and all the different areas and what's going on, I wanted to talk a little bit about brain plasticity and neuroimaging. So how we know what we know about the brain, the different ways that we've done these studies to figure out what does what, and how we know that how the brain changes, plasticity, okay? So the first thing we're going to talk about is studying the brain. Traditionally, in our more historical history, we learned about the brain before we had imaging techniques or functional recordings. We learned about the brain through brain lesioning. So these are really can be either naturally occurring, so this would be some sort of brain trauma. So your book has lots of examples of patients that have like two-letter names like HM, things like that. These are people that had really traumatic injuries to their brain and a piece of their brain no longer was there or no longer functioned. Or sometimes these are also brain lesioning can be induced. So there were situations, again, in previous medical history where, lobotomizing or cutting out pieces of the brain was thought to cure various things. And so we learned a lot about the brain in sometimes really terrible ways. And so this is kind of our historical view of brain development. A lot of what we knew before we had functional recordings or imaging techniques really came out of these brain lesioning studies. They're still super helpful. They're still super informative. We find out information about the brain through that process. But this is not something that we typically do now to find out more about the brain. Things that we do now are things like functional recording. This can be things like an EEG. That's the same kind of thing that you could have looked at your heart. It's the same kind of technology where we're measuring kind of that electronic pieces. We talked about neurons, and we talked about how neurons have this electrochemical signal. Because they have this electrical feature to them, to their processing, to their function, to their transmission, we can measure that. And we do that in a number of different ways. So again, EEG is one. In animals, sometimes we'll see single unit recording, and that's really where we put a probe basically into the brain so you can measure one neuron. And so our first research that we had that taught us about mirror neurons came from primates in these single unit recordings, where they had a probe injected to one neuron, and they would see that neuron would fire both when the monkey ate a grape or when they saw somebody else eat a grape. So again, we saw this information through those single unit recordings. These aren't things that are typically done with humans. They're more typically done with animals. Another one that's not as popularly used actually uses lasers. It sounds really scary, but near-infrared spectroscopy, NIRS. And it again does the same thing. It measures functional recording. Only it does it through blood flow in the brain instead of the electrochemical information. So it's still measuring function. It's just doing it a different way. So EEG, again, measures ERPs, which is our event-related potentials. So this picture here is a woman wearing an EEG cap. And what you see on the far right is an ERP. So you can see the actual event-related potential. And again, this is doing this through electrodes, and it's again measuring that neuronal activity. So this is that, remember we saw that video of the electrochemical signal going down the neuron when we were looking at myelin? It's measuring that. It's measuring that electricity that's in our brain. And it gives us really great information about how quickly we process things. So because the electrodes are on the outside of our brain, outside of our skull, outside of our hair in this cap, this very stylish and fetching cap, I might add, we get really good information about how quick the brain processes. So in this functional recording, how quickly something is done. So key presses, we'll see a video where it's just responding to different kinds of things. But the problem is because the electrodes are on the outside and are on kind of just like, it basically takes a cross section and it just measures there's electrical processing going on somewhere in this cross section of the brain, and so it doesn't give us great information about where this processing is happening. So we don't get great spatial information about where these things are happening. We get really good speed information, but our pictures are pretty terrible. They're a series of lines, so we don't have great spatial information there. NIRS, again, measures blood flow of the brain as an indication of function. Again, it's going to be great for temporal information as well. It does it a little bit differently. It's a little bit less invasive, so it's something that we can do better with children and infants. But again, it's going to have terrible spatial information for the same reason. We know that information is happening somewhere in this general region, but we don't know where in this general region that's happening, because again, it's kind of like taking core samples, where we just taking a core out and saying, okay, well, there's activity somewhere in here, but we're not exactly sure how much and exactly where it is. So again, we can get really great information on function, and that's what we call these functional recording techniques, because we get lots of information on function, we get lots of great temporal information. Now, if you want to get good spatial information, if you want to get pretty pictures, you need to do brain imaging. The most traditional ones that we see used are PET scans, MRIs, fMRIs. Rarely will you see x-rays used in psychological research, sometimes a CT scan, but mostly in psychological research, we see a lot of fMRIs. An MRI and an fMRI are very, very similar. The main difference is that F, right? That F stands for functional. So instead of just taking still pictures of the brain, it's a little bit more like taking a video. So again, it's not that simplistic, but it kind of is doing that. So we can both look at function and pictures of the brain. So again, instead of just getting still shots of the brain, we can actually look at the function of it. So here's what a PET scan looks like. It measures the metabolic activity using radiation. Because it uses radiation, it has a greater risk attached to it. And we know from our ethics research that that's not great. And so it's something that we have to make sure that we're managing appropriately. Pros, it's got much better spatial resolution than any of the functional imaging, the functional techniques that we were talking about. But again, it's really invasive. You have to drink this really gross stuff and then they measure it. And it also doesn't have great temporal information. So it doesn't tell us how quickly these things are. The pictures are better. But also it's important to note that PET scans are a lot older technology than MRIs are, and especially than fMRIs are. So fMRIs use magnets to record changes in the oxygen while the brain is active. So again, it's a little bit like that NIRS where we're looking at blood flow. We're looking at the oxygen in the blood to see where things are active. If it's a part of the brain that's active, it's going to get more blood flow because it's needing to be used, right? And so what we get are these really, really cool pictures, these really, really good images. We can get very precise. They're kind of like heat maps. So the different colors here indicate different levels of activity within those regions. Now the cons are that we got really bad temporal information, okay? These pictures, you don't know how quickly things are happening, the timing on things. Again, we get the best information from those functional recordings. And the other thing is that you have to be very, very still. If you've ever had to have an MRI of your leg or any body part at all, you know that you have to lay very, very still. So it really has a lot of problems to work out with children. So we can't really, while it's not invasive and it doesn't have these negative effects with kids, good luck getting a kid to sit still. I've heard of parents that have had to have their children, their young infants, have MRIs for medical reasons, and you have to bind a baby to do that. So that's not something that we really like to do in psychology then, because again of that harm. Babies really don't like to be bound very often. And so again, that can cause a problem. I posted a video in iCollege that will show you some of these different techniques so that you can see kind of what these look like. Next, we're going to talk about brain plasticity. So brain plasticity is the idea that the brain can be altered or changed. So think about plastic, right? You can mold plastic into anything. 3D printers are functioning on this idea that you can print something in 3D, anything out of plastic. So plasticity is this idea that the nervous system is adaptable, that it can be changed. And there's two different types that we're going to talk about. The first one is modifiability. And this is that functions or structures can be attuned. Okay, so needs to happen really early. So we have different parts of our brain. I know we haven't gotten to this different pieces yet. We have different parts of our brain that do different things. But if they're not getting the like the area that functions for vision is not getting vision information because the baby is born blind, the brain's not going to be like, oh, well, you know what? I'm not going to use that real estate. Whatever, too bad, so sad. No, the brain is totally going to use that real estate. And it's something else will take over, okay? So that part of the brain will be attuned. It'll be modified to be used somewhere else. So an example of this is in those congenitally blind infants. When you look at Braille reading adults, so these are infants that have never seen. And these are individuals as adults are reading Braille. They're seeing parts of their visual cortex. Again, they've never seen anything. They've never gotten visual stimuli there that are active when reading Braille. So that has been taken over by these other sensory systems. That real estate's been taken over. So part of the brain had been modified. A function or a structure was attuned to different needs. Again, this needs to happen really early. This is somebody that didn't have that vision information. And there's going to be sensitive periods for this modifiability to happen. The other kind that we're going to talk about is compensation. And this is when cells substitute for each other and enable recovery. So this would be something like after a stroke, after a traumatic brain injury, something like that. And so an example of this is that we see deaf will show the activation of the auditory cortex when presented with visual stimulation. So we're going to see kind of a switch over there. But this is really not unlimited. And it really depends on the extent of the damage, the age that the person is when they're damaged. The younger they are, the more compensation abilities we're going to have. We're going to see a lot more brain plasticity in younger individuals than we will see in older individuals. And it also will depend on the timing of this kind of need for plasticity as well. Other things that really affect brain plasticity are your genes. So different people will have different kind of genetic kind of predispositions to have more plasticity than other people will. We'll also see that experience will play a role. So different experiences that we have can change our brain. We know this just from lots of human studies, but one of the first really concrete studies that looked at this to the extent that this experience happened was Greenberg and his colleagues. And what they did is they looked at rats and they put two groups of rats. They had a group of rats in a really, really boring, sad cage. And they put another group of rats, and these were little young rats, in a really cool cage that had all these like rat tubes and all these really cool rat toys and all these different environments. And what they found is that the rats that had, this is for both younger and older rats, the rats that were in those enriched environments had heavier, thicker cortexes. They had larger neurons, which means they were healthier. And those neurons had more dendrites. Because they had more dendrites, they could make more connections with other kinds of more synaptic connections with others. And again, this was found for both the younger and the older rats. So this is, again, this idea that our experience can change our brain. Now, what's really interesting about this study, and is often less reported, is that there was a follow-up study where there was a third group of rats added. And they had the non-enriched boring rat cage. They had the super cool, souped-up, fancy rat cage. And then they had a third group of rats that were in a situation that really simulated their natural environment, what a rat would live like in nature. And what they found is the rats that had the more natural environment had even healthier brains than these enriched, souped up, fancy rat cages. So we really see that kind of from the evolutionary perspective, that the brain has evolved to develop in these natural settings to its highest potential. We also see that brain damage and other deficits can play a role in how plasticity works as well. And we'll see that there'll be compensation by other parts of the brain and other sensory functions. So you always hear that if someone loses one sense, other senses are heightened. And this is true because this accounts for this plasticity in our functionality. Okay, well, that ends our discussion there. And again, make sure you watch that video on the brain imaging. Thanks.